NLDA-SciChat for BioTech. Watch NLDA-SciChat talk about peptide-based vaccines. Find novel binding patterns and validate them in real time. Let's accelerate drug discovery. Chat mode, talk about peptide literature using fine-tuned LLMs with medical datasets. Let's start asking it some basic questions about peptides. Nice. It gives a verbose answer based on existing scientific literature. Now let's get more specific. Let's ask it about peptides and MHC receptor interaction. Now instead of giving a guessed answer, it is actually asking you to run a simulation. Simulation interface. Call it from chat. Select peptide. Run simulation. Just type Start Simulation Interface. Here you can see the detailed interface given by SciChat for the simulation. You can select peptides, receptors, and binding sites using the given drop-down boxes. You can even choose to run a single simulation or screen multiple peptides at once. For now, we will run a single simulation for our peptide interaction with the MHC2 receptor for easy demonstration. This usually takes some time depending on your compute resources. It gives us a detailed answer based on the simulation. It acts like an interpreter of the results. But for confirmation, we also need structural analysis. You can see it has given a very decent delta G value for this peptide. You can also look at the binding pose of the peptide in the protein structure. But is it really enough? Now imagine running 1,000 simulations. It will take a huge amount of time and resources. SciChat will automatically solve this with a math equation. This equation can act as a surrogate for following simulations. Now let's ask AI to find a mathematical representation of the binding pattern. Now why to do that? Right pointing arrow. If we can find an equation, we just need to do a calculation, instead of a whole long simulation, to pre-screen promising candidates, which actually deserve to be simulated, hence, accelerating and democratizing the discovery process. As you can see, it has found out a math equation found using 30 binding points. We will validate this equation using a simple Python script by comparing to known literature. See the millisecond benefits of math and Python. Here in the logs you can see the generated equation being used. Known literature for a peptide, GRGDS's binding affinity. And what our equation predicts it to be. As you can see, it just produces a 1.39 kilocalories per mole error margin for this one. Not bad at all for just 30 curve points. Imagine what if we had 1,000 plus binding points. You run a 1,000 point simulation once, the rest is handled by the equation. Yes, even on a low-end laptop. Now, let's add a second layer of scrutiny, just to be sure. We will do a comprehensive web search for literature using Claude Opus 4 analysis of the equation. Disclaimer. This is just a test. Never upload your findings on any AI cloud if you care about IP safety. As you can see, a lot of the points are validated for the SciChat's generated equation. It even suggests a relationship between the parameters not known in existing literature. Now, how cool is that? We just found a novel mathematical representation to calculate binding affinity. Now by using this equation, we can pre-screen promising candidates. Instead of repeatedly running 1,000 plus long simulations for peptides and MHC, we just found an efficient relationship between them that can help us pre-screen thousands on candidates even on a basic laptop. The result? 50 simulations of pre-screened candidates instead 1,000 random seeds each time. The impact? A whopping 80% saving on your resources. All from a causal chat interacts. NLDA-SciChat, accelerating drug discovery. DM for early access today.